should your Gimsa, or if you pronounce it Gimsa stain look like? Your red blood cells should be uniform and lightly pink. Your background should be clean. Parasites, when they're present, have sharp red chromatin dots, clear blue cytoplasm, and they are intracellular, not floating. Looking specifically at the stages, for your early ring, you have a delicate ring. You will get 1 to 2 chromatin dots. The chromatin dots may actually appear as dark purple. This is the parasite nuclei. Often you will get 1 to 2 rings per red blood cell. The cytoplasm is thin. You get that pale blue ring. And the central vacuole gives it a headphone look sometimes. Then when you reach your late ring to early trophozoite stage, you get thicker ring or a body, and this means more cytoplasm, and the pigment may begin to appear as fine as brown granules. For the mature trophozoite, you usually wouldn't see this in peripheral blood in humans, but when you're culturing, you will see it. So these have more cytoplasm. They appear coarser, so the pigment tends to be a bit coarser. The contrast between the parasite and red blood cells cytoplasm is less with Gimsa than Leishman. Rare in peripheral blood, but if you're culturing, then you will see these. Get more cytoplasm and so coarser pigment appearance. The Schizont typically sequestered in humans, so you wouldn't see these, very rare. When they're present, multiple merozoites within one red blood cell, but when we're culturing, we expect to see these. Now, the gametocytes, these are characteristic crescent or banana-shaped parasites, filling distorted red blood cells. These are dense chromatin with scattered pigment granules. So that brings us to the end of this video. Thank you so much for checking out the video. I really appreciate you subscribing to our study group and giving the video a thumbs up. And I'm looking forward to catching you in the next video. God bless.